today. Yeah, thank you for letting me speak here, and I'm very excited to explore with you like a new project. It was like a side project. We were just trying to like, can we also like go over the limits of the RNA polymerase and see if we can also incorporate other modifications. And then we were surprised, oh, it works quite well. And then it turned out to be like a nice side story. And I will explain you that one now. So first of all, we all know that RNA therapeutics is getting more and more popular. We have the mRNA vaccines, we have ASOs, small interfering RNA, as we have just heard, and then also aptamers. But one problem is if we make them out of RNA and then inject them in the body, they get degraded quite quickly. So therefore, what can we do? We can modify the RNA. And luckily, there are many different positions where we can modify the RNA. So we can either like change the phosphate backbone, we can use alternative ribose triggers, or we can modify the 2' end of the sugar. There's also like a different position where we can modify the RNA, like in the bases. And then on our lab, we also do like base modifications, but this you already heard like on Monday from Julian. I'm interested now in increasing the stability of the RNA. And one modifications we also use in our lab is, for example, the 2' fluoro modification. Yeah. So we have the 2' hydroxy group replaced with the 2' fluoro modification. And this is already shown to increase the stability. And we normally use these in all pyrimidine bases. But then we can also exchange the ribose with another ribose trigger to also like increase the serum stability. And there we have, for example, the LNase, binders, or TNase. And I want to focus on the LNase. And already Julian introduced it nicely yesterday why we want to use the locked nucleic acids, the LNase. So LNase, they have like an extra methylene bridge here, and this keeps it in the endoconformation. And first of all, increases the stability against nucleases, so the resistance. But secondly, also increases the melting temperature from like a strand when you have a DNA, like an LNA DNA or an LNA RNA strand. So this can also be like some nice features which you can give like a higher stability, or you can expand like in a larger space of structures which you wouldn't have without this modification. And they're already like also quite used in many promising therapeutics. But how do we get these LNAs into the RNA? And what is already like known literature, you can see a lot of DNA polymerases which were engineered to incorporate these LNA modifications. And often like they start with a DNA primer or like an RNA primer, and then they incorporate these with also quite high efficiencies and low error rates. This is quite nice, but when we want to have like an RNA helix and we don't want to use the primer, then we have to find another way. And additionally also, these engineered DNA polymerases, they also like hard, not as available as when you have like polymerase, which you can also commercially obtain. Then we were thinking, we already know that the T7 polymerase mutant with like one mutation can incorporate 2' fluoro modifications, which we already like use in the lab very regularly. Why not try to see if we can also incorporate the LNA modification? And to test this, we designed a library where for the aptamer selections, but we focus that we don't have any modifications in the constant region, so in the primer binding sites, because we were like, we don't want to challenge the polymerase like in the beginning of the transcription, because the initiation, we want to get the transcription running. And secondly, also we're interested in the random region to see how well it gets incorporated, the LNase, and therefore we don't want to bias in the beginning. And thirdly, also the increased thermal stability could also be a problem then in later stages in the helix when we do the reverse transcription and we have the LNase in the primer binding sites. So I performed the transcriptions, and we had, like as I just mentioned, we had two libraries, one for when we incorporate LNA modifications. So then we have the LNA, and you can see it works fine when we have the specific library. But if you then have the library where we have the A's in the primer region, then we didn't got a transcription product. The same is when you use LNA TTP, where we then have the specific library for the U modifications, we get a transcription product, and where we don't get, when we use the other one, we don't get one. So it showed also actually that the library design also was quite important, that we don't have any modification for primary region. But we recognize and see, okay, we can get transcription, but how good is this transcription? Because, okay, we can get like an RNA product, but is it actually also like a library? Do we only get transcription of all the sequences without the LNA modifications, or do we get like a bias in that? So therefore, I transcribed the RNase, reverse transcribed with the superscript 3, which already was also known to read LNA modified RNA, and then performed next generation sequencing. So first of all, I looked at the sequence, the nucleotide distribution. So we had like first also a library which had 25% of every nucleotide. And after this one round of transcription and reverse transcription, you can see that when we use LNA ATP, that we actually got an increase of T's. And if you look at the LNA TTP, then we got an increase of A. So this was the only bias. But when we then look at the LNA modification, so LNA ATP, we actually have like roughly the similar amount of A's compared to the C's and the G's, which we're looking at. So it's actually quite nice to see that we actually have like 20% of LNA modifications in this library. So it was already something we could work with. Another thing also I was interested to see, okay, how is it when we have 2, 3, 4, or 5 LNA modifications in a row? Is this also possible? And then this data set, I was just looking also at the repeats. So here we have the base repeats, which just means we have 3 repeats of the same base in the library. And they can also see that you can actually also incorporate like multiple LNA modifications in a row in the library. So we actually have quite a high sequence diversity, which is also important for science. But then I also was interested, okay, we can get like a nice library, but is it actually also like the transcription product which we want? So if we have like a template, we want to have like that the RNA has exactly the sequence, because otherwise we could lose a lot of interesting aptamers. And therefore I used then 12 randomly generated sequences, but this time I knew the sequence. So then I could go in the sequencing data set and could say, okay, expect this base, and I get this base in my next generation results, and then could calculate what is the fidelity. And you can see here on the left, you can see the 12 sequences, which I chose. And on the bottom, you can see the substitution patterns, which I then observed. And one trend you could see was that for the LNA ATP, we had like a higher error rate than for the LNA TTP. But in general, when we compare them to the controls, which I have to mention also, we use 2'-fluoro CTP and UTP for these. That's also like because we always use that in selections. We got an increased error rate, but compared to also what is shown in the literature for the other DNA polymerases, that these error rates are still like also in a moderate nice low range to work for selections. And with these informations, we were like, nice, why not try to do like now a selection to see if we actually can do like multiple cycles of transcription, reverse transcription, and then also actually get like a aptamer. So therefore we use the LNA TTP modification because these are the lower error rates, it has a higher yield during the transcription. And yeah, and therefore I chose the target, which also was used during my PhD, is the influenza virus, and specifically the hemagglutinin protein. It's a surface protein which is binding to your cells and is needed for the viral selling. So therefore I got a his-tagged protein and performed the CLEX, and here just want to show that we did first a counter selection so that we don't get any aptamers binding to the his-tag, and then we did several rounds of selection. After 4 rounds of selection, already like I did sequencing because I had other selections also to sequence, and then I was like, okay, just like have a look, how does the library compare? And I was quite surprised that already after 4 rounds of selection, I had actually only 2 sequences which were dominating the pool by 90%. So in the beginning, I saw that and was like, okay, that doesn't seem like there would be some binding because it seems too fast for an enrichment. But then actually, I ordered these 2 sequences and also the runner-up. And when I did then some biolay endophthalmometry studies, I could actually see they were actually binding quite strongly to the target. So we have like a low nanomolar range affinity for both of these sequences. And the aptamer 1 has 3, and the aptamer 2 has 7 LNA modifications, which you can see here. So we have like here a small cluster for the LNAs, and then for the aptamer 2 we have like 7 LNA modifications together with the 2'-fluoro modifications which we also use. And then I was curious to see, are these actually important for the binding of these modifications? So then I transcribed these aptamers, but then substituted, for example, the 2'-fluoro CTP with the CTP and the LNA TTP with either DTTP or with UTP. And you can actually see for both aptamers, when I remove these modifications, so when I transcribe them with the unmodified substitutes, then we lose all the binding for some of the sequences, or we have like a much lower binding. And this shows that it's actually important to do the selection with these modifications already in there, and that you can sometimes lose the binding of the aptamer, like when you afterwards want to introduce these modifications. And that was quite nice to see. But then also the big question is, I was claiming that the LNA modifications increase the stability of the aptamer. So therefore we also did a serum stability test, and we could see actually when we have both modifications, the aptamers are stable for over 4 days. That when we stop the experiment, would be actually interesting to see like how long can we actually go on. But when we removed one of the modifications, we could actually see that immediately a drop after minutes of this stability, so we lose the full length aptamer. And yeah, and then recently, just like in the last weeks, we actually got then also some nice results and could actually then also already see get a cryo-EM data of the aptamer. Because now it's interesting also to see like, are these LNAs also actually maybe interacting with the protein, or are the LNAs only important for the binding? This is only like very preliminary, so this was like initial screening, so these are only 150 micrographs, so we need like a lot of more data. But it's already quite interesting to see that we already can see the aptamer and the yellow on top of the protein. And with this information, hopefully we can also then use this aptamer and design structures to then have like a nice inhibition of the influenza virus. And it's not only like the only protein which we targeted. He also was like doing in parallel another selection targeting the CD40 ligand. And there actually also you saw like only like after 7 rounds in enrichment. So it was not only that like with the LNA libraries, we got only like after 4 rounds a really huge enrichment. In another selection with another target, we actually got like a much later enrichment. It was quite nice to see that it was not like too much pressure on the selection. But if you want to read more, I didn't have the time to talk more about this protein. So in conclusion, we could actually see that commercially available RNA polymerase mutant could be used to incorporate LNA modifications. And you could also like select with this polymerase and with this modified library then aptamers, which are very highly serum stable and have like a high affinity to the protein. And we hope that we can use that maybe for also some aptamers, maybe even like in challenge conditions where you have like an increased temperature, where other libraries would fail, but then thanks to the LNAs, we could select some aptamers which like a higher thermal stability. And with that, I want to thank especially Laia, who was like also co-author on the paper and who also like helped a lot with the analysis, and also Jörn and Julian for the supervision and all the other collaborators, and you for listening.